Welcome, everybody. We're just waiting a moment for the full number of participants to join, and I think we're just about there. So thanks, everybody who's attending today's webinar. I'm Steve Gourlay, the CEO of Actinogen, and I'm pleased to introduce to you our Chief Medical Officer, Dana Hilt, who's here in Australia helping us with the analysis, who's based in the United States. And then Dr. Steven Targum MD, a world expert in psychiatry trials, published more than 170 papers by the looks of his resume, and a very experienced assessor of depression and other psychiatric trial data. So welcome, Steve.
Thank you.
Today we'll present to you the top-line results of our phase 2A proof of concept trial of cognitive impairment and depression. And the top-line result is that we have clinically and statistically significant Xanamem benefits in symptoms of depression, which is a very exciting finding, essentially underlining that we have an effective, potential novel new antidepressant with a new mechanism, and there is no other drug being developed that has the same potential to use this mechanism in the brain for depression. But we did not meet the primary endpoint of a cognition attention composite, in large part due to a very big placebo effect, which will be explained to you. A bit of housekeeping. We will save Q&A to the end of the discussion, so the presentation will take about 25 minutes, and there'll be plenty of time for discussion in the next 20 to 40 minutes. We will make the presentation available. It takes some time to load onto our YouTube channel and the website, so that's likely to be available late in the day today. If we don't get to all of the questions, we will endeavour to answer them by email or in communication through our email shareholder distribution. So with that, let me also move to the disclaimer slide. As usual, we will be making forward-looking statements. The details of this are in the presentation published on the ASX today. These slides are all and in the same order with the same slide numbers as in that presentation on the ASX this morning. And so now I'd like to introduce Dr. Dana Hilt to present the next main results slides.
Okay, thank you very much, Steve. So I'm happy to go through the data of the trial, and then we'll have some discussion. So to refresh your memories, this is an outline of the depression trial, the XanaCIDD trial. So this was a phase 2 double-blind proof of concept study, and this is the first study that Xanamem has been used in patients with depression. And so the patients have their diagnosis confirmed by psychiatric interview, and have residual depression. So these are patients that still have persistent depression, and they can either be yes on an antidepressant, a stable antidepressant, or no, previously treated with a stable antidepressant, but not presently taking them. And they are then randomized to either the active or the placebo, treated for 6 weeks, and then followed up for an additional 4 weeks for a total trial length of 10 weeks. The primary endpoint, as Steve has alluded to, is what we call attentional composite using the CogState, which is largely a computer-based platform to assess cognition, and it looks at attention and working memory. And then the key clinical endpoint, frankly, is the depression scale, the Montgomery-Åsberg Depression Scale, the so-called MADRS scale. This is a scale that virtually all drugs for depression are measured in clinical trials for approval by regulatory agencies, the FDA, the EMA, the TGA. So that is the most important clinical outcome measure. And then there are secondary measures of cognition and additional measures in the study. And we enrolled 167 patients, and they're equally distributed to the active or the placebo in a blinded way. Then the blind was maintained through the entire trial. Next, please. We go to the next slide. Yep. So as we think about going through the results, then the trial was well conducted. The data was excellent. There wasn't very much, or there was very minimal missing data. And this trial was enrolled in Australia and the United Kingdom. So one of the things one wants to look at is to ask the question of whether the results were different based on the country, or were the results different in high enrolling sites versus less high enrolling sites. And we did not see any obvious significant differences there. So that gives us confidence the data is excellent data and really is a good measure of what we're trying to assess in the study. First of all, the drug was safe and well tolerated, and this follows the theme based on the previous studies. And we now have more than 350, 400 patients treated with the drug, and it has been very safe and well tolerated. And I'll get into some of those details in a few minutes. But there are no serious adverse events related to Xanamem that have occurred in this trial. And in fact, there haven't been any in any trial to date. So I think we have assembled a pretty large body of data now to tell us and inform us that the drug is safe and well tolerated. Now, importantly, on the clinical endpoint, which was depression, there are clinically and statistically significant treatment benefits of Xanamem. We'll go into that in detail. And they are seen either at the end of the treatment period, which is 6 weeks, or even at the follow-up period of 10 weeks. And I'll be commenting on some of the observations that we've made that the positive effects we see with the drug seem to continue for that 4-week period, even after the patients are off treatment, suggesting the drug may have had some impact on underlying biology, positive impact. Now, as far as cognition, there was no treatment benefit on the attention composite. We think that this is largely attributable to the fact that there was a large, unexpected, and unprecedented improvement in cognition of about 0.4 points on the scale that we're using versus 0.3 points Xanamem. And so this was a very large, unexpected placebo effect, and we'll show you that data in a few minutes. Next, please. Now, to think about or to comment then on the effects on depression, that Xanamem treatment performed better than placebo, and the magnitude of the effect is clinically significant. And in many cases, in many of these analyses, you'll see it's also statistically significant. In all patients, all 165 patients that had data at the end of the trial, a trend towards benefit was seen at the 6-week visit and was actually statistically significant, clinically significant at the 10-week time point at 10 weeks. That in the patients who had less severe depression, and this is measured by the MADRS scale, and the MADRS scale, the higher the score, the worse the depression. And so the median here is about 26. So if you look at the patients who still had significant, clinically significant depression, but not as severe as the most severe patients, those patients actually had a clinically and statistically significant improvement were seen throughout the study and at 4 weeks and at 6 weeks, the end of the treatment period, and at 10 weeks. And so that's a pretty substantial effect. I'll show you those data. In the patients who weren't taking antidepressants, and remember, we enrolled 2 groups, patients that are on stable antidepressant therapy or patients that previously had a good 6-week trial of an antidepressant but were not on antidepressants during the study. And patients can elect not to take antidepressants for a lot of reasons. There are side effects. There are a variety of side effects that patients do not want to experience. But all the patients had residual depression. In that smaller group of 31 patients, there was a very substantial treatment effect in depression with Xanamem treatment. Next, please. Next. So a bit of a housekeeping and background methodology slide. These are some of the methods that were used in the trial. So the primary statistical methodology used is the so-called MMRM, mixed model for repeated measures. And this is the standard, gold standard measure to look at data, repetitive data during a trial to see whether there's a benefit at the end of the trial versus the beginning of the trial. This is the type of methodology that the FDA, the EMA, the TGA really like the sponsors to use in clinical trials. The secondary endpoints, including depression and the secondary cognition endpoints, were analyzed in exactly the same methodology. Importantly, all of the analyses I'm going to show you today are pre-specified. This is not post hoc trying to find a group that responds. These are all pre-specified, we had a good rationale to think that these groups of patients would be patients that might respond to the drug. So all of the analyses today that you'll see in this presentation were pre-specified and followed a very strict statistical analytic plan. The p-values are one-sided hypothesis testing. And one of the things you will see here that is used widely in neuropsychiatric trials is a metric called the Cohen's d. Okay, so the Cohen's d is a portion of the standard deviation as far as a treatment effect size. So if you look at the physician's desk reference, we use it in the United States, these are all approved drugs. All the approved drugs have a Cohen's d of around 0.3, 0.25 to 0.3. In the drugs that are approved, for example, in Alzheimer's disease that have procognitive effects, those drugs have a Cohen's d of 0.17 to 0.25. Many antidepressants have a Cohen's d of 0.25 to 0.3. And so that will allow you to look at the Cohen's d's, which we put on the graphs I'm going to show you, and put it in perspective as to how does this treatment effect compare to other drugs and to all of the approved drugs that physicians use, which is about 0.3. Next, please. At baseline, then, we enrolled a group of patients that were on average 49 years of age. It was more females than males, which is not surprising. This is depression. And at eligibility, we used a separate scale. And this is a good clinical trial hygiene method to try to decrease variability, is to use scale A for eligibility, but then scale B to measure the treatment effect. So we use the Hamilton Depression Score, valid, widely used scale for depression, to assess patients for eligibility and baseline, and then we use the MADRS scale at baseline and thereafter to determine whether or not the patients had an antidepressant effect. And what you see is that the groups are well matched, 21 in both groups on the HAM-D and 24 and 26 on the MADRS. The fraction of the patients that are on background therapy is 75 to 80%. We did see a little bit of difference in the baseline attention composite. You'll see in the placebo, it's minus 0.1, and then in the Xanamem, it's plus 0.1. And that may possibly have had an impact on the analysis of the primary endpoint of the attention composite. Next, please. So if we go through the data now, get down to the details, we'll look at all patients, all 165 that had data, and we're looking at the MADRS change. Now, the MADRS is an inventory of depression symptoms and signs. And so the higher the score, the worse the patient. And so what you want to see here is go down will be an improvement in the patient. And on this graph, the dark purple line is the treated group, and the gray line is the placebo group. And what you see is the treated group is starting to separate from the placebo group. And at the end of the 6 weeks, there is a separation with a Cohen's d of 0.24 and a p-value of 0.1. And then if we go to 10 weeks, now we see that there's an even larger separation with a p-value of 0.02, which is significant, and a Cohen's d of 0.43. Okay, and so over time, there's a gradual separation of these groups. And interestingly, and I'll point this out on other graphs, is that after the drug is stopped at week 6, and then we have week 10, many of the metrics, many of the measures in the patients that are on Xanamem show a persistent or even improvement in week 10, whereas the placebo patients are reverting back towards their baseline. So we see this systematically in many of the measures that we conducted in the study. And we think this could, in fact, be an implication or suggest that the drug is really having some more fundamental impact, not a curative, but a positive benefit on the underlying brain function structure. And this is something we're going to follow up in the future. Next, please. And if we look at the subgroups that I've talked about, and these are all pre-specified, the left graph is measurement of depression in patients who are less severely impacted at baseline. And here you see a very clear and obvious separation of the treatment group, Xanamem versus placebo. Again, down is better. And this is significant at the end of the treatment period with a Cohen's d of 0.88, which is, if that could be repeated, that would be quite substantial, and a p-value of 0.02. And then at 10 weeks, a persistent benefit, again, statistically significant separation. If we look on the right, these are the patients that with more severe depression at baseline. They don't really separate. Interestingly, though, at week 10, after the drug is stopped at week 6, and then we continue to monitor the patients, you see the expected deflection up in the placebo group, and you continue to see some improvement in the Xanamem group. Next, please. And if we look at the two different groups, patients on antidepressants on the right, patients not on background antidepressants on the left, and remember the group on the left is small, and so trying to achieve a statistically significant p-value is difficult with only 31 total subjects. But you do see a clear separation with, again, down is better, the Xanamem treated group doing better than placebo, and at week 6, the Cohen's d is 0.6, which is very good, and the p-value is 0.06. Obviously, if these groups had been larger, that delta would have been statistically significant. And there is small groups, and you see some convergence at week 10. The right panel is patients on background antidepressant. You see a gradual separation at week 6 with p.2, and then a greater separation at week 10 that's significant with p.02 and a Cohen's d of 0.51, which is pretty solid and robust. And so we're seeing this antidepressant effect in a number of different groups, the overall patient population, patients not on background antidepressants, a little stronger, but still on background antidepressants, a gradual separation, which is increasing at week 10. So this multiple streams of data then show us that the drug does have an antidepressant effect in these patients. Next, please. Now, turning to the attention composite, what you'll see here is the unexpected placebo response, and I'll spend a few minutes going over this. Now, at baseline, again, the key here is the dark purple is Xanamem, and the gray is the placebo. Now, on cognition, up is better, okay? So the opposite direction for orientation. And you can see at baseline that there's a difference between the groups, that even though this is a pretty reasonable sized trial of 165, 167 patients, randomization didn't completely do its job insofar as there's a little bit of separation there, and we don't know the impact that had on the overall analysis here and how it impacted the measurement of cognition. But you see a pretty dramatic increase in cognition throughout. Now, to put this in perspective, in previous studies, the placebo response is about 0.15 on this attention composite scale. So if you look back at baseline at the light gray line, you see that they started at minus 0.1. And so the dotted line is where they should have maxed out during the study. Well, you can see that they certainly went well in excess of that. And this is something that we think greatly impaired the ability of this trial to detect the expected procognitive effect of the drug. Because when you have a placebo effect that's so strong, it is almost impossible to detect a treatment effect. It washes out the treatment effect and decreases your ability to detect it. So I don't think we can actually definitively state that the drug doesn't have a procognitive effect. It didn't on this measure, but the point is, with this robust placebo effect, it's very difficult or impossible to measure a procognitive effect. Okay, so that is, I think, one of the reasons why the drug did not show this effect on cognition in the study, even though it has had procognitive effects in a number of different studies previously. Next, please. Turning to safety, the drug continues to be safe and well tolerated. We can see a list comparing the Xanamem treated patients to the placebo treated patients. The fraction that had treatment-emergent adverse events during the trial is similar. There were no serious adverse events in the Xanamem-treated patients. There was one in the placebo patients. There were a couple more patients with treatment-emergent adverse events leading to discontinuation or interruption or pausing of the drug, but none of these were serious. And if we look below at the incidence of treatment adverse events that occurred in greater than 5%, you can see that headache actually occurred more frequently in placebo than in the treated group. And the rest of these are pretty close in frequency, fatigue, and URI-like symptoms. So again, the patients in this study with depression show a very excellent safety and tolerability profile, highly acceptable. And we think that's very much consistent with what we've seen previously in the normal volunteer studies and in the previous Alzheimer's study. And I think, yeah, maybe I pause here, and if Dr. Targum, Dr. Gourlay, any comments on or additional elaboration on the depression results, and then I'll spend just a couple of slides talking about the top-line conclusions and the implications for our ongoing development.
Sure. So I can pick up here.
Yes.
And I'm going to repeat everything Dr. Hilt said, but I'm going to say it from Boston, largely. First of all, I had a chance to look at this data last Friday, and a few things I want to say about it. And I'm only going to focus on the depression part of it because that's my area of expertise. These were really 3 studies going on simultaneously. If we can go back to slide 10, we'll look at the primary study, which is the study of 165 patients. Is it possible to go backwards? Yeah. It's a little bit easier to follow that way. So here we have the large study of 165 patients. And again, we're seeing here a 1.5 point difference at 6 weeks on the MADRS. It is interesting and curious that the patients who were receiving the candidate drug continued to improve a little bit and the placebo group deteriorated. All of these patients knew after week 6 that they were off medication. So as Dr. Hilt suggested, it might be, and again, it's only a suggestion, there might be some biological effect that is sustained beyond the 6-week period. It raises questions about the length of treatment that is necessary to get a true antidepressant effect. But again, a P of 0.11 is nothing to write home about at this point in time. Now, there were 2 other studies that in some ways are even more compelling. Can we have the slide 11? And this is the study of what we might call the mild versus the moderate depressed patients. There were 81 patients in the group with MADRS scores of less than 26. And in that population, we see at week 6 a significant difference between the candidate drugs, Xanamem and placebo, of 0.02. It's a small sample size. Let's remember this is a phase 2A proof of concept study. But as humans, we are always looking at the efficacy as if it's a fully worked out phase 3. So you have separation here, which is largely sustained through week 10, even though the patients are no longer taking the drug. In the larger sample, we see that the Xanamem group continues to improve beyond week 6 into week 10, raising again this question. And it's a question whether or not there is some sustained biological effect going on beyond the 6 weeks of treatments, and what is the correct amount of time that a patient should be on this drug to show a true drug effect. So this is the second study, the severity study. The third study in slide 12, if we can move to that, is also compelling. And here we have what I would distinguish between a monotherapy study, that is patients who are not on background antidepressants. And in this study, it meant although patients met criteria for major depressive disorder, met criteria for treatment resistance, they had been on no antidepressant for 6 weeks. So they were coming into this study essentially on no drug, no antidepressant. Alternatively, the background antidepressant, what we call augmentation studies, includes a group of patients who are on a variety of antidepressants, a variety of doses. They may or may not be taking those doses, We don't know how long they've been taking them. We just know that they are taking an existing compound we call a background, and now we're augmenting it with a second drug. They are essentially two very different populations. Most people involved in clinical trials would agree that a monotherapy study is a lot easier to do because there are fewer compounding variables and the patients tend to be a more homogeneous group. And here we did see, again, that substantial difference, a Cohen's d of 0.64 at 6 weeks in the group who were on monotherapy. In the background antidepressant group, we see what we saw before, a sustained benefit going on beyond 6 weeks to week 10. So what does this mean? Data is data. It is a small study. It's fascinating to see the dissociation between the cognitive effect, where we had a higher than anticipated placebo response, and the antidepressant effect, where we got pretty much a placebo response that we'll see in most clinical trials. So again, interesting. It's data, and I think it warrants more consideration. Thanks.
Yeah, thank you, Steve. Steve Gourlay, Dr. Gourlay, any other comment, or we want to continue on?
Look, I think it's probably good to move forward to the
Okay.
conclusions and implications. And I know we've got a number of questions coming in, but
Yep.
please be patient and we will get to those. So we've got plenty of time.
Okay. Okay, so then again, to reiterate some of our conclusions, then the drug was safe and well tolerated, and the safety profile is very consistent with what we've seen in the prior studies. We've gone over the data for both the cognition and the depression. We think the reason the drug did not achieve a separation on the primary cognition endpoint, the attention composite, is because of this large and unexpected placebo effect, and we're starting to explore that and try to figure out what happened and why this was. And we are just, importantly, as we think downstream or in the future, does this apply to the Alzheimer's study? And I'm going to get to that in a minute. We're using a different set of attention and cognition measures in the Alzheimer's trial than we're using here. So it's a very different metric, a very different methodology is being used. And as far as the depression, the most important clinical measure and endpoint in this patient population, there are clinically, in many cases, in many of these analyses, statistically significant treatment effects, and some of the Cohen's D's were quite robust and large, seen in MADRS both at the end of treatment and this persistent, interesting effect 4 weeks later at week 10. And we think that persistence of benefit beyond the time in which the patient has been taking the drug, merits further investigation. It may suggest, as both I and Dr. Targum said, that might be some underlying biologic modification in the patients. And when we think about this mechanism, it's quite different than the standard approach to depression. Standard approach to depression is basically altering neurotransmitter levels. This I think is very different. This might have a more fundamental impact on some underlying biology than just the manipulation of the neurotransmitter levels. And want to emphasize, first of all, that all the analyses we've presented here are pre-specified. This is not post hoc. We will be, these analyses represent sort of the top 30 plus tables and data sets, and there's another 150, 180 to come from the data analysis team, and we'll be looking at those. We do not think that additional larger data set will in any way impact the bottom line conclusions from this, but it may help us investigate certain things, may help us investigate further the placebo effect on cognition and add more details to the interpretation of the study. Next, please. Okay, and finally, what are the implications of this for the Alzheimer's study? Well, I think this is another test yet of this drug that it actually showed that it gets into the brain, can have a positive effect on brain function, and can have a positive effect on depression in this case. Now, it's important to think about these two trials and determine whether there's any translatability here. First of all, the depression trial is a short trial of 6 weeks. We're looking at symptoms, reversal of symptoms. that the Xanamem, the Alzheimer's trial, is 9 months, and we're looking at a slowing in the rate of progression of the disease. So we think these are really apples and oranges. These are two different treatment scenarios. We're asking the drug to do different things in completely different groups of patients. So, we don't think that a lack of a pro-cognitive effect in the depression trial will translate into the Alzheimer's study. And the most important measure we're measuring in the Alzheimer's study is the CDR sum of box measure, which is a clinical functional measure, which in our first AD trial, we showed a benefit in. The other point I want to make is that the ongoing study in Alzheimer's disease now is a confirmatory trial. We did an initial 2A trial, and that was recently published. You can access that on our website over the last month. And in patients who had a clinical diagnosis of Alzheimer's disease, but also had biomarker positive proven Alzheimer's disease, we showed a pretty significant clinical benefit in that subgroup of patients. And so the Alzheimer's trial is confirmatory, whereas this initial depression trial was more exploratory. It's a 2A, was a 2A study designed to ask the question, well, is there an effect in patients with depression? And we think we've established that there is an effect, positive effect. And then finally, there is a pretty substantial set of preclinical data in Alzheimer's models to show that 11-beta HSD1 inhibition can work. There is not a very good array of preclinical models for depression, so there isn't the corresponding type of preclinical data for depression. Next, please. And so this is just to review what we're doing now in Alzheimer's disease. And again, this is a 9-month study, not a 6-week study. So it's a much longer study. Now, the study is designed to enroll 220 patients that have a clinical diagnosis of Alzheimer's, but also have a positive, what's called a p-tau plasma elevation of p-tau 181. This confirms a diagnosis of Alzheimer's disease. And we know that in patients that have a clinical diagnosis of Alzheimer's and plus have the elevation in p-tau, that those patients are going to progress more rapidly and they have bona fide Alzheimer's disease. So that's who we're enrolling in this study. The total study is 220 patients. The primary endpoint will be 7 cognitive measures. We're not using the same measures in this trial as we used in depression. We designed these measures to be particularly suited to this patient population and to the type of cognitive effect we expect to see. But the key clinical endpoint is the CDR sum of boxes, and that's a functional cognitive composite that has been used with most drugs that have been approved for Alzheimer's disease. And this is the endpoint that the FDA and the EMA and the TGA and other regulatory authorities feel comfortable with. And this is the one they want you to do to get your drug approved for Alzheimer's disease. And we're also measuring a real life measure of activities of daily living, the so-called Amsterdam ADL scale. Now, the way we're conducting this study is we've opened up a number of Australian sites. We're going to enroll the first 100 patients in Australia. And then after those patients reach 24 weeks of treatment, it'll be active and placebo. We'll do what's called an interim analysis, a mid-course look at the data. And we have yet to decide on the details of that. Will remain blinded, will be open. What we'll want to see is that the data is headed in the right direction. And then at that point, we'll enroll the balance of the 220 patients. And those patients will be followed for a total of nine months, which we think will be an adequate period of time to measure the decline in the patients, given that we're enrolling patients that have positive p-tau biomarker. Next, please. And I'll give the speaker's staff back to Dr. Gourlay.
Thanks, Dana. And yeah, just to add to what you said, we are adding US sites currently, so we may well enroll quite a few of those first 100 patients also in the US, Dana. So just to maintain our enrollment timeline. So just want to finish off with a couple of slides, really showing our pipeline. It is unchanged. these exciting positive top-line results for depression in our depression program were something we'd always hoped for. We chose cognition as the primary endpoint because we had more data on cognition from our previous healthy volunteer studies. But obviously, in retrospect, depression would have been better. And in future studies, the focus will definitely be on depression rather than cognition as a primary endpoint anyway, for sure. So our Alzheimer's phase 2b, as Dana said, is ongoing. We're enrolling in Australia currently. We'll be enrolling in Australia and the US shortly, and aiming to have and on track for data with an interim analysis in the middle of next year. And as a reminder, we do have another important asset, Fragile X program. We have an open IND and an approved phase 2 protocol. We chose to pause that program to focus on the 2 lead indications because we had so much data emerging on our ability to impact positively patients with Alzheimer's and patients with depression. So that's still an asset when it comes to partnering discussions. We are interested in the future exploring other types of dementia and perhaps cognitive impairment associated with schizophrenia. But for now, we're focused on our top 2 indications being Alzheimer's and major depressive disorder. And now last slide, just where are we with all of these programs? As Dana said, interim analysis middle of next year for Alzheimer's with final results about a year later. That's a very important focus for us, and we are working hard to enroll good sites, and there's a lot of enthusiasm out there for a drug like XanaMIM, which is a once-a-day, safe, and we hope very effective oral medication, much easier to use with a much better clinical and commercial profile than these new injectable antibody drugs that are only modestly effective. Subsequent to the interim data, a positive readout there would encourage us to open a second pivotal trial, which we would call a phase 3, in parallel with the final stages of that trial. Our phase 2a depression trial, we've just given you the top-line results. We'll continue to analyze the results in the coming weeks with the final full data set. We go straight into partnering and potentially FDA and EMA discussions, the regulators, around agreeing on pivotal trial endpoints, which would go into our next phase 2b/3 trial, which would be focused on depression as the primary endpoint. And then hopefully a second phase 3 would be potentially doable in parallel. We have ancillary studies to complete, or you always have to do clinical pharmacology and some additional non-clinical studies, particularly for a depression program. And we have a number of other publications in the pipeline. And so to summarize, I'm very excited by these data. It's a pleasant surprise that we had such a big potential benefit on depression. And it's quite evident that this is a very strong basis to go forward to discuss the future of our depression program with both partners and the FDA. And it also has an exciting read-through to Alzheimer's disease in that this is yet another clinical trial showing clinical activity of the 10 milligram dose, meaning it's getting into the brain, it's inhibiting its target, and that is having a clinically meaningful effect. So in some ways, it means the cortisol hypothesis is validated by these data, and we look forward to being able to publish this in a peer-reviewed journal and at academic meetings in the near future. So with that, I'm going to pause and invite people. Sorry, I forgot one more slide. So just to summarize, I think I've already said this slide, but evidence of XanaMIM activity in depression is very encouraging for the whole program. And the persistent antidepressant effects really do suggest, as Dana said, activity on the underlying brain processes. And so we will continue to work both on the depression program and the XanaMIM phase 2 in Alzheimer's disease. We currently have a cash runway through late 2025, having recently raised some money with the existing funds and also accrued funds from our expected large tax rebate and other sources. So, and there's a couple of references at the end of the deck. So I'm going to stop sharing at this moment and refer to the Q&A. And Michael, if we need to share slides, you might need to do that so that I can do the Q&A.
I can will do, Steve.
The first question is from Alan Soren. Hi, Alan. You say that this cognition result has no implications for cognition in AD, Alzheimer's disease. If so, why was this trial conducted?
So you start, or I should start?
No, Dana, you feel free.
Okay, yeah. I mean, look, we don't have time here, but there's a very solid biological rationale why blocking cortisol in the brain is beneficial to patients with depression. So we did this trial because we thought we could help patients with depression, and we did. So that's the bottom line.
Yep. Thanks, Dana. Dev has got a question. What is or was the rationale for not using MADRS as the primary endpoint rather than COG state attention composite? So Dev, the honest answer is that we went back and forth on MADRS versus cognition. And because there was, a wealth of scientific rationale for cortisol as a mechanism in depression, but no interventional credible, there was marginal and credible, information, but not the same as our own randomized trial evidence for cognition itself. So we chose to go with the clinical science, but we obviously made MADRS the key secondary endpoint. Question from Ashley. The results look very marginal, with the placebo group getting very similar benefits to our wonderful drug, which is that is true in cognition, but certainly not in depression, as we showed. Why does the announcement say the results are positive? The market clearly disagrees. Look, I'll just answer this one. Hopefully now that you've seen the presentation and heard the commentary, you agree that the results on depression are clinically significant and really form the basis of being able to take this program forward as a novel antidepressant with a new mechanism of action, which is a huge scientific discovery, let alone obviously very important for the future of the company. So I think it's a technical study that needs to be explained and understood by people who are not as in the same field as ourselves technically. So that would be my answer for you, that I think we will do our very best to explain this as clearly as we can, but our interpretation is that this is a huge positive result for the drug, not just for depression, but it also reads through to potentially a very positive result in the Alzheimer's study. Alan had a question about placebo, but it was a bit tongue-in-cheek, so I'm actually going to skip over that, Alan. Well, I'll say what it was. Does Actinogen have any plans to market a placebo for cognitive enhancement? And no, we don't.
That was very informative.
Right. Right. So Mark asked a question about why was a one-sided p-value used? I don't know, Dana, would you like to answer that? Or
I can start and you can, yeah. So
sure.
At a phase 2A trial, lots of times a one-sided p-value is used because you're trying to ask the question, is your drug better than the placebo? You don't want to discard a drug that is better. For confirmatory studies, obviously a two-sided test will be used. But if you look at some of these p-values, they're less than 0.025. So even a two-sided test would have been positive in those endpoints. I don't know, Steve, but if you have additional.
Yeah, thanks. No, that's exactly right, Dana. I mean, Mark, if you want to you can just do a two-sided test by doubling the p-value. So 0.02 equals 0.05 under a two-sided test. So that's easily done for interpretation. Mark's got a few questions here, so I'll try and do them quickly. How would you set up a follow-on study in MDD? What changes would you make? Dana, I'll let you maybe.
So I'll make a couple of comments, and I'd like Dr. Targum to comment because he has a wealth of experience. I think, with this persistent effect, I think we want to do a longer study, and we'll definitely want to have a follow-up period. So I think doing doing a study for 8 weeks, and many depression studies are 8 weeks, but 8, 10, 12 weeks is probably advisable based on the type of biologic signal we're seeing here. I think we want to study the full set of data to go back and determine exactly which patient population we want to enroll, where there's this big difference between patients not presently on therapy and on therapy, whether the more severe or less severe patients. That's all going to inform, I think, the design of a study. I know, Steve, what's your thoughts on that?
Well, I certainly agree that a longer study is warranted. 6 weeks is, for this particular compound, may have been short, particularly if it is, and like very broadly calling this disease modifying, but this is a modifying kind of agent. Going back to the earlier questions that we're talking about statistical significance, given the small samples in the sub-studies, the antidepressant on or off, the MADRS high or low, the very small samples, I think the effect size analyses are really much more telling than a statistical significance for these studies. And so obviously we're going to have to create enough power in future studies and decide what exactly the criteria will be for depression severity, for previous history of antidepressant use, whether it's monotherapy or augmentation. And one other thing I'm thinking is, cortisol's been a topic of interest for depression for over 50 years. In the 70s, we were publishing about dexamethasone suppression tests and non-suppression, and thought we had a marker for depression. It's interesting that you didn't do that here to see whether or not there was a subpopulation that actually had cortisol impairment associated with their depression. So that's another thing to think about going forward.
Yeah, I mean, Dr. Targum's alluding to the fact that I remember this from medical school days, but it's not done anymore, where if you look at a patient with depression, you think, well, their cortisol level can't be elevated because cortisol's a stress hormone. They're sitting there, they're withdrawn from interaction, they're quiet, they're depressed. But if you actually give a normal subject dexamethasone, which is a very potent corticosteroid, it suppresses the brain's systems to generate cortisol, and it doesn't do that in patients with depression because they have a high level of cortisol already. So I think we have a lot to think about here and digest and decide, how does that inform a next study in depression? And then one final comment to emphasize what Steve has said. Most phase 3 trials with depression using the MADRS use per group 150 to 200 patients per group. Okay, they use more patients per group than this entire study to get a p.05 and an average MADRS delta of 2.9 in the last 20 approved drugs. So, it's too early to look at this data and say that the signal is greater than that, but then in many cases, the signal, in fact, is greater than that in this present trial. So we have to do a larger trial and really try to refine the target patient population.
So let me say one more thing so everyone gets their continuing medical education credits here. In the 70s, when we were doing dexamethasone suppression, approximately 40% of patients who met criteria for moderate to severe depression failed to suppress their cortisol on 1 milligram taken at 11 p.m., in contrast to essentially none of the controls. And the reason that pretty much died as a marker was because of the early development of drugs like fluoxetine, which worked for everybody. And so people were less interested in confirming that someone had a depression than simply putting them on an antidepressant. And so it really wasn't continued. But the interest in cortisol has not died out at all.
Yeah. And also, you can't admit patients to hospital and do a dexamethasone suppression test. No insurance company will pay for it in the United States. So, it's not something that's done routinely now.
All right. Thank you both, doctors. Just keeping an eye on the next 10 minutes so that we can finish on time at the top of the hour. Mark, I'm going to skip over your last question because I think we answered that already about antidepressant effects. John asked, was the magnitude of the improvement in attention composite in the XanaMem group, say 0.3, in the expected range? And the simple answer to that is yes.
Yeah, right.
In fact, 0.25 to 0.3 is exactly what we saw in the active group in our cognitively normal volunteer trial. So
Yep.
yes, so our drug did improve cognition in exactly the way it was expected, but as we saw, placebo did too. David asks, so what do these trial results mean for possible future cognition endpoint trials and other indications, specifically schizophrenia? So Dana, maybe I'll let you answer.
Well, again, schizophrenia is a very different situation than depression. The schizophrenia cognition trials, ones I've done, have been 3 to 6 months in length. And I think that's, if one were to do a schizophrenia cognition, the so-called CIAS studies, they would be longer. You know, placebo effects on cognition, there have been some reported in very shorter Alzheimer's disease studies, very back when donepezil and rivastigmine were first studied. They do tend to wear off. You know, placebo effects are not persistent forever. So if you do a study that's longer, usually those will abate over the course of a month or 2 months. And so, you know, the CIS trials are typically 3 to 6 months in length.
Okay. Thanks, Dana. I'm going to skip ahead to a question from James because it was related to the last question about the effect size on cognition. If the placebo group had behaved as expected, say it goes up to 0.05 and our drug had gone to 0.3, would the results have been statistically significant? And actually, James, that's exactly how we powered the trial.
Yeah.
So the simple answer would be yes.
Yes.
David asks, could you summarize how potentially effective XanaMem could be compared to other MDD drugs? I mean, based on what we've seen today, is it similar? Is it better? Do we know? Or do we need to do another trial?
I think the profile of the effect is different than most antidepressants. I wouldn't hazard a projection, a guess, or not more an estimate this time. I don't know, Steve, what's your thought on that?
Well, depressive disorder is a heterogeneous disorder. And so some people are going to respond to rapid-acting antidepressants, do just great. Plenty of people respond to SSRIs, but 30% of patients don't respond sufficiently to anything. And so there are a group of patients who are resistant to existing treatments. You still need new agents, and this is a very unique mechanism.
Yeah, there's unfortunately a very large population of depressed people in the world. So
yeah, and the overall antidepressant market is massive. It's 17, 20 billion dollars, and yes, as Steve Targum said, not well treated in a substantial proportion of the population. Graham asked a slightly different question. How did you arrive at the dosage amount? If increased, do you think the results would have changed? And maybe I'll put my clinical pharmacology hat on there. So I think the team before Dana and my time as well, and a big shout out to Bill Kettleby, former CEO, hopefully other former folks at the company are listening in, but they did a great job of getting a human PET study done with 40 older people, showing that doses of indeed 5, 10, and 20 had high levels of binding and interaction in the brain, and in particular 10 looked like an excellent dose because and 5 is a possible efficacious dose. So and when we've looked at our cognition data, 5, 10, and 20 in cognitively normal volunteers had exactly the same effect on cognition. So the same is true of hormones in the body. So we actually have a lot of information guiding us to use doses of 10 or even less. And in some future studies, we will have a 5 milligram dose group as well. So to answer your question simply, it's very unlikely that 20 milligrams would have been any better.
Yeah. And those studies are published and you can access them via our website.
Yeah. Thanks, Dana. Tim asks, says, congratulations on the results. Would you consider bringing in a partner for depression, i.e. phase 3, independently of the Alzheimer's treatment arm? That's a great question. Generally, because our drug XanaMem is just a very small 10 milligram once a day pill, got our to be marketed formulation being used in the Alzheimer's trial. It's generally not regarded as possible to split one disease from the other with a different pharmaceutical company for each. That's typically not done if you have the same pill and the same dosage formulation. So no, we would probably, well, we will be talking to partners about potential global partnerships to help speed drug development. Obviously, they would contribute intellectual and clinical development resources as well as money into the program under that circumstance. And we may well consider regional deals where their contributions would be to do some regional drug development as well as contribute financial resources. So deal making is definitely on the table for the next 6 months while we continue to talk to the FDA, develop the next trial, and implement the Alzheimer's trial. Nathan, I'm going to skip over your comment, but thank you. Richard asked, how do these results impact your strategy repartnering and your opinions on how these partners will look at the results? I think that the partners are all waiting eagerly to see the results, and I think as soon as we've got the data complete and ready for that in the next week or two, we'll start discussing that with them. So I don't really want to say how they will interpret it. I think they will be very intrigued by positive data on depression with a new mechanism. I don't know, Dana or Steve, if you want to comment on that particular thing.
Nothing in addition to what you said, no.
Okay. Good. All right. Well, I think we're pretty close to the top of the hour. So we've answered virtually all the questions in the question bank. So just left to me to say thank you to everybody who's participated. Thank you for those of you who tuned in to listen. We're very excited by the results. This is really clinically meaningful. It's actually, in my view, a really important scientific discovery that cortisol matters. And back to Steve's comment about 50 years of scientific literature and history, this is the drug that gets into the brain that actually tests the cortisol hypothesis. And positive findings here are a really big deal academically, scientifically, and clinically. So and thank you, Dana and Steve, very much for joining on your Sunday afternoon in the US. All right.
Thank you.
Thanks, everybody.
Thank you.